All right, it's 12 o'clock. Welcome to this week's Iowa Learning Farms Conservation Webinar. I'm Matt Helmers with Iowa State University, and Director of the Iowa Nutrient Research Center and faculty affiliate with the Iowa Learning Farms. It's really my great pleasure to introduce our speaker today, Dr. Francois Brigand from North Carolina State University. For those that might have been on last week, we got to hear from Dr. Danny Winterley, and she had been a student in Dr. Brigand's lab in North Carolina State, or at least an undergraduate student at North Carolina State University. And so we are really fortunate to have him with us today. He's a professor and university faculty scholar there in their biological and agricultural engineering? Okay. Yes, every department, a little bit different. And so he's going to talk to us today about are we losing a lot of nitrogen during the denitrification hot moments in agricultural soils? With that, Francois, I'll turn the virtual floor all over to you, excuse me. And if people have questions, please type those in the chat box as we go, and we'll get those to Francois at the end of the talk. With that, now the virtual floor is all yours.
Thank you, Matt, for this nice introduction, and thank you for inviting me to give a talk. So I propose to same slide here. Are we losing a lot of nitrogen during denitrification hot moments in agricultural soils? I want to obviously recognize my co-authors, because as you'll be able to see, it's not a one-person job or the result of one-person work here. Dr. Chad Poole, who's extension specialist in our department, assistant professor, and Miss Nora Sawyers, who did a lot of the work on the data that I will be showing. So some of you might have seen some of these slides. I'm trying to put things together and be a bit thought-provoking. So it's probably less of a extension talk than a research talk, although there's an extension component in it, as we will see at the end. So let's make sure we're all on the same page about what hotspots and hot moments are. So there's been 2003 a very important paper, seminal paper by McLean and others, who said, well, there seems to be hotspots and hot moments at the interface of terrestrial. Vidon added a little bit of a subtlety about what these are. And so what are they? Well, hotspots seem to be patches that show disproportionately high reaction rates relative to the surrounding matrix. And hot moments, which I will abbreviate with HM, are short periods of time that exhibit disproportionately high reaction rates relative to longer intervening time periods. And this is what we're going to actually focus on today. And are there such things as the denitrification hot moments? So, I'm sure a vast majority of you, if not to say all of you, know that there are hot spots for nitrogen retention. And in most watersheds, nitrate is the prevalent excess inorganic nitrogen form, particularly in our agricultural watersheds. And yes, denitrification is considered to be the most significant removal mechanism. And what are denitrification hotspots? Well, how do they work? Well, they need saturated conditions where the electron donor, that is the organic matter, is present, and where nitrate becomes the electron acceptor. So these are a bit of a jargon here for us, but as long as we can create anaerobic conditions where there is a source of carbon, organic matter, and we put nitrate in it, the nitrate should be filtered out. And yes, there's been 40 years of research that looked at the riparian zones, and particularly I like this graph. This is in Normandy in France, so I thought I'd bring some new landscapes for you. And there's it's been clearly shown that from the field edge to the stream edge, the nitrate concentrations tend to really decrease, and it's been shown quite nicely over the years. And indeed, the nitrate load is greatly reduced. In fact, after 40 years of research, we know a whole lot more about the nitrification and its drivers. This has been, in my opinion, this discovery in the mid-80s of this role of riparian buffers has been provided at least an enormous leap forward in our understanding of what's happening in the watersheds. We know that riparian buffers remove about 30% of the nitrogen they receive. And again, this is kind of the number I like my students to remember. But however, to this day, you know, we've had a drain mod, we have a VFS mod, vegetative filter strip mod, but there's no such thing as a riparian buffer mod, an RB mod. After 40 years of research, you know, drainage, we had a model after maybe 20 years. You know, I'm one of Dr. Skaggs' students, so ex-students. So it's kind of weird that we don't have, but there's no clear effect of the vegetation type, the width of the buffer, and I mean, a lot of the research has been focused on, you know, the soil type, vegetation, width, and all that stuff. But that didn't lead to anything able to that we would put in an ensemble that say, okay, here's our model. So to me, these are really indicators that there might be something big that we have missed. So I will stop here on that. Now, I'm just bringing you another piece of information, which is nice observations that my colleague Dr. Chad Poole has made. First of all, in North Carolina, yes, there's no doubt, no farmer out there denies that something is changing. And particularly, we have both, you know, droughts and floods. And guess what? Nobody would have thought so 10 years ago, but clearly irrigation has become a need in North Carolina. This is some data. So I know this is not very, very clear. I just took almost the raw data. This is some water table depth that you can see it's going up here. And look at the month of June and July, it's nothing. There's just no water. And not only this, but when we look at the matrix potential during the month of June, that we're going in a really, really low zone where, well, essentially there's just no water available for plants. And that's a big problem. And yes, that is new. And for that reason, Dr. Poole has actually started taken advantage of houses and the drainage plots that were established by Dr. Skaggs and Dr. Gillum in the 80s and late 80s and early 90s. And he's actually conducting all sorts of trials to test water management scenarios, but also hybrids and all that. So he's done that for corn and soybean. So that just gives you an idea. So this site has been established for at least 30 years. And it's lots of great things have been done on this. So I'm just going to go on to show you. So yes, we have 8 houses where an entire water management system, including sub-irrigation, has been put in place. And, you know, where you have a main, we have structures to maintain source pumps, and we also have laterals to actually sub-irrigate. So anyway, my point is not to talk about this, is to show you some of the data that are really intriguing. And so, yeah, Dr. Chad has actually, you know, tested wet versus overdrained versus optimal conditions and looked at what were the consequences of that particularly on yield. And yes, some of you who have done irrigation for decades will say, well, welcome to the real world, North Carolina. We do know that when we are too dry or too wet, we are losing yield. And yeah, but, you know, for us, it's new. And so it's been very important that Dr. Poole has been actually able to show that. And yes, he's tested different hybrids, and this is, you know, hence the 20 and 17 numbers here. But that's beside the point what I'm trying to do here. So yes, guess what? Yes, there are, in any dry or wet stress, there are loss of yield, and that actually translates in actual real money numbers. And that's obviously, as we all know, the farm scale, that's really the bottom line. And that drives everything, and rightfully so. So what about the nitrogen fate? And this is, I'm going to show you, this is a little bit small here, but I want to show you that, so the intensive drainage is on the left here, and optimal is in the middle, and poor drainage is on the right. We'll come back to the right later on. As you can see, actually, in optimal conditions, what goes to the grain, that is in green here, is actually higher than what would be in intensive drainage. And actually in intensive drainage or too dry, actually there's still a lot of nitrogen uptake. In fact, very close to that of the optimal conditions, but a lot of the nitrogen actually stays in the stover. Okay, so now what are these wet conditions I'm going to try to talk about? And in the wet conditions, I just want to show you, these are water table profiles that have been measured in the wet, that is blue, and as in optimal conditions in green, and free drainage in black here. And if you look at what's happening with the wet conditions, there are lots of water table variations, ups and downs within the first foot of the soil where the organic matter is concentrated. And I'm going to introduce this concept of draining and re-wetting cycles. Indeed, when you look at this, you have a drainage when the water table recedes and sudden re-wetting following a rainfall event. And as you can see, there are many of these happening in these conditions. Now, when we looked at some of the nutrient balance that Dr. Poole was able to conduct out there, what he found is that there is essentially enormous amount of unaccounted for nitrogen. So 84 bushels per acre or 90 kilograms per hectare lost in 90 days. This is enormous. So something very special is happening. I mean, we tried to run a drain mod N, man, we could get maybe 30 kilograms per hectare in 30 days, but in 90 days, but no more than that, and certainly not during the growing season. So third concept that I want to introduce is these hydrological hot moments. Is there something that's happening during weird times? Absolutely. Well, hydrological hot moments and the pulse nature of hydrology. I'm sure the vast majority, not to say all of you, have already seen what a hydrograph looks like. We have a baseline and we have some storm flows. And I've actually purposely taken a watershed or at the stream order 3 so that we can have actually some baseline. But I'll come back to a drainage system later on. And so one can actually take this very data and sort it from the highest to the lowest. And the same hydrograph now looks like this, right? So it's the same data from the highest to the lowest. And from that, we can actually calculate volume duration curve. How do we do that? We actually integrate under that curve, and we create that corresponding increased red curve here. So at the end of the day here, we can actually express the cumulative flow volume here in millimeters corresponding for 100% of the data, but we also can express that as 100% and percentage of the total flow. And this is what this curve is. So what this shows is actually the same data, but instead what I've done is I've changed the scale here. That's a QQ plot scale. So all it does, it actually shows, highlights what's happening in the first, 2, 4th, and 5th percentile of the time. And these are the corresponding values. And what this shows is that it shows that in 15% of the time, corresponding to the highest flow values, we have about 52% of the flow that occurred in that particular watershed. So now I'm going to go back to a more busy slide where this is actually drainage values, or rather from a drained agricultural watershed. And now what this shows is that in 10% of the time, corresponding to the highest flows, right, or the hot moments, we have 55% of the flow that occurred, the flow volume. And for nitrate, we had more than 70% of the load of nitrate that occurred in 10% of the time. So it just tells you that in very short moments, in these hot moments, corresponding following rainfall events, big things are happening. So we need to start thinking no more in terms of time, but in terms of these fluxes. So anyway, so that tells me that something very big is happening, or it's highlighting that during these hot moments of hydrological hot moments, what I call hydrological hot moments. All right, so majority of the volume of drainage water is exported in a small proportion of the time, and a majority of the nitrate load is exported in a small proportion of the time. So now the question is, and additionally, in poorly drained soils during hydrological hot moments, the water table is in the organically rich part of the soil, as we saw before, and the pulses of flow and nitrate are actually accompanied with sudden increase in water velocity in the soil. So water is just not nicely going like this during these hot moments. Actually, the reason why we can have a lot of water exported during that time is because water is moving much faster because the hydraulic gradient are much steeper at these times. Now, the real question is, okay, well, if that really defines a lot of what's happening in the export, what happens to the biogeochemical processes during these hydrological hot moments? And this is where I'm bringing another totally different piece of information that we collected over time, which are the lessons from WICHE bioreactor studies. And Dr. Helmers was a part of that study that I'm going to report here. So, I'm not going to be able to go back to what WICHE bioreactors are. And at one point, for sure, to some extent, I certainly thought these were the perfect nitrogen removal hotspot, just like a riparian zone. But this time we actually could maintain the anaerobic zone all the time, which is not the case of what's happening in riparian zones. So, of course, we take advantage of the fact that anaerobic conditions are going to occur. And as a result, we can actually, the nitrate becomes the next more powerful electron acceptor, and then yes, nitrate is chemically filtered out and leaves the nitrogen leaves the aqueous system in gas. Okay, but our WICHE bioreactor is so perfect. The theory says it should be. The practice shows that, well, after 2 to 3 years, there often is a decrease, a removal efficiency from 60% maybe to 20%, and maybe this. Some of you might say, well, this is a bit of an exaggeration, but there's a trend towards a sudden drop. So we did this project where the ideas was, well, one of the ideas was, could one find a way to rejuvenate these bioreactors, make them more like riparian zones or denitrification walls that seem to work all the time? And so we tested the hypothesis that maybe these draining, rewetting cycles I just talked about, you just go up and down, you just saturate the system, drain, saturate, drain, might enhance the denitrification. So for that, we actually ran, this is what I do for a living. So we ran a pretty high-tech experiment. And this is where we had actual WICHE columns here where water was coming from the bottom and was leaving. We were actually testing for both water and gases. And the water, we actually tested it thanks to a high-frequency water quality sensor and created a sampling system that would sample sequentially from each of the columns here, just 1 or 2 things here. So what we tested over almost an entire year, almost 300 days. We said, okay, well, we're going to have these columns saturated all the time, but we're going to give it 8-hour breather for just some of these columns, and the others are just going to stay saturated. And so again, weekly, 4 of the columns were just got a breather. And then when we got these results, okay, well, we need to look at if the duration of the breather matters. Is we give a 2-hour breather, 8-hour breather, 24-hour breather. Does that really matter? And yes, how did we do all this? Yes, we actually created this sampling system, which allowed us to actually obtain at the end nitrate and DOC concentration every 2 hours. And we also did that for gases. And at the end, we had hourly data. But so this is kind of the secret of this thing is of this sampling system. And we'll come back to that later. Well, it turns out that, minutes, so these rectangles here correspond to the breather, and the rest is the week that was going on. So the saturated conditions stay saturated. They are the blue ones, and the columns that had draining and rewetting cycles are in yellow here. And we're looking at essentially the denitrification and removal rates per meter cubes of bioreactor per day. And within minutes, this is the important part, minutes after rewetting, the denitrification rates are just multiplied by a factor of 2 to 5. As you can see, it's happening. And why is that happening? Well, it seems to be actually stimulated by the release, at least the one we could see, the apparent release of good organic carbon or dissolved organic carbon. So in fact, we could create a correlation between pretty darn good correlation between the release of DOC and the removal rates. Anyway, so at the end, did this breather help? Well, heck yes, they did, and these are a lot of the data that you can see here compared to the draining rewetting cycles. And yes, it does wither over time, but for the first 1 or 2 days, clearly it's quite enhanced. And what did we find between for the duration of the exposure of the aerobic conditions? Well, yes, 24 hours really created much higher denitrification or stimulated denitrification to the point that in fact we could at the end there was almost a linear increase of the denitrification ability with the increase of the duration of the aerobic conditions. So that's one thing. The second part of the hydrological hot moments, so that just tested whether or not we go up and down in the water table in agricultural soils. The next part is, well, yes, these variations up and down are also accompanied with a vast increase of flow, velocity, and concentration. So this is where we actually use the same system, but in a horizontal bioreactor, in the lab. And what is it that we found? Oh, so how does this work? Well, we actually adding water from one end, and water just goes from left to right here, and we can adjust the outlet. And we have ports from 1 to 12, and we can actually see what happens to the concentrations as the water dries. And there are the things we're measuring temperature and dissolved oxygen, and we are able to actually collect the water physically with our fraction collector. And here is our actually, water quality sensor. It's just spectralize it from scan. And from that, we're able to obtain, again, very high frequency data. And this is the type of data that we're able to obtain. So this is one here is actually the inlet and 12 is the outlet. And so as you can see, the outlet concentration is like this, but as we actually, these are real pulsing concentration. So at the inlet, we're trying to have a low, quote unquote, low base flow concentration. And then we suddenly increased it also during a flow pulse. That was also a step-like function like this. As you can see, obviously the nitrate concentration is not coming out very high on the first pulse, but at the end, it's getting there. Yes, it's lower than the inlets, which is a good thing. But anyway, all that, so we analyze all that data, all these data, and then what we found is that when we had short both flow and concentration pulses at the same time or long flow, we essentially multiply the removal rates by 2 to 4 during the pulses. And so it was like a big aha moment, because we're thinking not only are we actually creating wetting and drying cycles, but while this is happening, water is actually moving much faster in the soil, or in this case, which is bioreactor. And that too stimulates denitrification. So there's a different levels, that additive level. So what we found from these bioreactor studies is that, well, the effective soil reactive volume is multiplied by 2 to 3 as the water table itself rises into the water to the soil surface, possibly. Number 2, during a rewetting cycle, because of the fact that we just give a breather, and as a result, we actually release a lot more carbon that feeds the nitrification. We're multiplying everything by 1.2 to 2.5, and the actual pulsing itself due to increased velocity. So what it does, actually, we think, it actually activates more pores, and it creates large, at the micro scale, it creates larger fluxes of nitrate from the middle of the pore towards the substrate. All that to say that, my hypothesis is there must be hot moments of denitrification during hydrological hot moments in agricultural soil, at least for sure on a wet soil here, because there's a clear evidence that there probably are hot moments of that. And just using our data here, and I agree this is just bioreactor, and that's why it must be tested for actual soils. But we're not talking about an increase of several percent or maybe 40%. It's a factor of 8 to maybe 30. Again, these numbers are maybe inflated. I don't know. I can't tell. But it's not small. It's by several fold for sure. And the question is this the reason for this loss, especially extremely high loss that we couldn't account for? So if this hypothesis were confirmed, to me, there are two conclusions that are important. On the research side, denitrification models are clearly ill-suited to take into account these hot moments, because these denitrification models are based on the idea that for a given organic matter content, U and a given saturation, and temperature, this is what you're going to get. Well, in our example here, it's clear that for a given substrate, a given temperature, the denitrification rates were multiplied by a factor to 0 to 5. 1 to 5. 2 to 5. So obviously, all our models are ill-suited for that. If this is happening a lot of the time, and all the time, in fact, well, that's really problematic. On the extension side, well, then there are lots of things on the water management, recommendations that we have on water management and fertilization that must be revised, because if indeed there's a lot of fertilizer that we actually add in lumps, well, and we have all these hot moments of denitrification, well, maybe we're unknowingly losing maybe, this is not 80 out of 220. That's not negligible. That's 25% of what you just added. Whoop, that's gone. So I propose some different consequences for research. I'm sure there will be other questions that will be there that will be quite interesting. And it seems to me that we must establish the conditions for when, why, where, etc. denitrification hot moments occur or not in agricultural soils. However, catching this elusive yet potential central process may be one of the next frontiers in water quality research. At least that's what I really believe. And, I actually believe that if there is a place where we can do that and have a chance to actually answer some of these questions, it's actually like artificially drained systems. And the main reason for that is because we have access to the hydrological functioning very well. Compared to a natural riparian zone or a riparian buffer, researchers have complained for decades they can't tell where the water is going. Really, there are just too many macropores and everything. In our case, the uncertainty is greatly reduced. Is it all gone? No, but it's greatly reduced. I don't think any one team can do that because it's hard. Number one. Two, we've done that as one team, and I've heard all sorts of comments which are rightful. Well, yeah, but it's because of your conditions, and maybe it's true, but maybe it isn't. And the truth of the matter is that if it's really a big thing, we must look at it together at many sites at the same time. This is hence my as a community here. And plus, it's hard to do, and there are techniques to do that we've maybe pioneered to some extent that I would love for us to share. Because if confirmed, hot moments apply to all denitrification hotspots in the landscape, and that means that in particular, maybe buffer zones, and might be one of the reasons why we just don't have a model that works. And in my opinion, one of the big consequences that our colleagues in the eco-hydrology world or the biogeochemistry world that's more less attached to agriculture, in my opinion, there's no chance of predicting the impact of climate change if indeed these hot moments are a real thing, because we do know that the hydrological hot moments are going to change for sure with the climate change. So some of the thoughts on the consequences for farmers and extension agents. Again, this is less of my forte, and I would welcome comments here. We must establish tools that will predict nitrogen losses to denitrification hot moments under well-defined conditions, and then we must update our water management and fertilization recommendations to least lose nitrogen, optimize nitrogen uptake by crops, and that goes to the grain. And that's, to me, that's an entire new world that we have in front of us. So the conclusions for me are actually very hopeful. What I'm saying is that if indeed, to me, denitrification hot moments are a great riddle in our understanding of the nitrogen cycle. Are they real, or is it just a bust? It happened to work on Wuxi bioreactors, but in reality, it has no chance to work anywhere else. There are too many confounding evidence, at least in our organic soils in North Carolina, where for sure there must be something that at least is related, or at least loosely to that. It seems to me that there's an opportunity for the next generation of water quality researchers, and these opportunities are immense. But they will require to work as a community because it's hard to catch the elusive process. And if only one team does it or too few teams do it, there will be, one of the reasons why buffer research was done and was successful is because everybody did it everywhere. And that's one of the great lessons. And one of the reasons why I'm a great admirer of this, all these teams that have been done. And we must do that in a pluri and transdisciplinary approach because at the end of the day we need to know what releases that carbon that generates the biogeochemical hot moment, and that is not for only amateurs or only amateurs of hydrologists rather, but it's for people who deal with the rhizosphere, the fungi people, the genetic people that do genetics, people that do organic matter, all that. So there are lots of other things. And with that, that will be my my conclusion, and I will take and yes, we need to have opportunities to recommend our recommendations to climate change. And this is thank you for your attention. Hopefully I wasn't too long. And I will welcome questions.
Okay, thanks, Francois. Let's go through those last couple slides, and there got time for people to write in questions. We have a few already. If you're on today and you are wanting to earn a CCA CEU, please send your name, the name you entered to watch the webinar, and your CCA number to Elena Whitaker, ALENAW @iastate.edu. Again, Elena Whitaker, ALENAW @iastate.edu by 5 o'clock today. Next slide. All right, if you watch this webinar live and are willing to fill out the voluntary demographics survey, we'd greatly appreciate it. Only one survey response for 2024 webinars is needed. You can go to iowalearningfarms.org/survey or scan your QR code. Hopefully I talked long enough that folks can scan that QR code. We'd greatly appreciate it. And again, only one survey response is needed for 2024 webinars. And let's go next slide. We will have Wenjun, she's on today. So Dr. Huang will be talking about, can diversified cropping systems help increase soil carbon storage? So another great talk, I think. And then maybe one last slide. Okay, there was somebody that asked about the PowerPoint. All these webinars are archived on the Iowa Learning Farms website. The address is there. Also, we have a Iowa Learning Farms webinars playlist on YouTube, so you can go find that. I think the recording will have worked, so we'll get it up there as soon as possible. Okay, we've gotten a few questions, Francois. The first one was they're in a pivot irrigated system in southern Alberta where they have very dry conditions, and asking, are we causing or promoting an end surge by irrigating every 3 days? A 1-inch application makes about a 70-hour full circle.
Well, yeah, that's a good question. Possibly. Yeah, I would love to have the answer to this. Again, I'm not convinced that these denitrification hot moments are happening all the time. And this is because, for the optimal conditions that we have, I didn't have time to really present everything, but the optimal conditions and the dry conditions, the water table were much lower. And in these conditions, I don't know if we're irrigating every 3 days, actually. I don't know that. But, the water table were much lower than they were below, essentially most of the time below 1.2, maybe 2 feet. And as a result, we did not see some really big unaccounted for nitrogen loss. So it seems that for the optimal conditions, which includes, irrigation, when needed, we didn't have that problem. Now, if you guys are irrigating every 3 days, no matter what, and you let water rise to the top of the soil, maybe. I don't know. At least that's my hypothesis. I hope I helped. Again, it seems to me that it's a water table variation, and whether we create saturated conditions within it and occur.
Yeah.
That's how I see it. But again, that's only that's my one view from the resulting from what I've done on the draining and reweighting cycles. But it's possible there are other things that are happening that we just haven't seen, obviously.
The next one, and this one was talking about the saturation fluctuation experiment, and nitrate removal was significant, and wondering, was that nitrate converted all the way to N2, or was there N2O production during those drying rewetting cycles?
Yeah, great question. Well, the good news was that, in fact, there was a lot less N2O production during the aerobic conditions. In fact, it seems like the N2O, the N2O that might have been produced was probably oxidized, rather, I was either oxidized or reduced all the way.
Okay.
So all I can tell you is that the gaseous part of the experiment did show that, yes, there's much less. I think it's a reduction by 80% of the N2O that was emitted under constant saturated conditions.
N2O.
Yes.
Yeah. Yeah. Okay.
So
Okay.
where did the nitrogen go? I don't know if it went all the way to N2. All I know is that there was a lot less N2O produced under draining and reweighting cycles.
Okay. There was a question, how did the flow rates compare between the saturated and drainage rewetting columns?
So the flow rates, they didn't change any. We kept the same flow rates all the time. So we were not trying to look on that part of the experiments. We did not change that. So now for the horizontal bioreactor, we ran weekly experiments, and for the last over the weekend, essentially, we drained the bioreactor and we re-wetted at the same time. So in our pulse effect, we had two things, but we were trying to get things that were somewhat similar and repeatable, and of course they were not entirely. But by draining, we created this draining and rewetting stimulation that we've seen in the column study. That's one. But then this is where we tested our pulses. So when we test our pulse, some of it is due to the draining and rewetting cycle, but yet the pulse itself did create its own stimulation. Hopefully that helps.
Have you done any work using biochar and whether that impacted any results?
No, I wish
Okay.
I wish I had done that. We do have an experiment with Dr. Birchall here, but it's about adsorption of ammonium and phosphate, so it has nothing to do with it. No, I haven't done anything with biochar. My understanding of biochar is that the organic matter in there is a lot more stable, and so I don't know if it would actually react to draining and rewetting cycles. But that's a great question. Yep. We would love.
Um,
If you want to collaborate, I would love to collaborate on that.
um, okay, did any other water quality attributes change with the hot moment experiment?
Well, the big one that we did test whether or not we had ammonium release, and we didn't do that systematically for all of them, but we did enough to use. And every time we did, we couldn't find any ammonium. Number one. Number two, the big one that changed, yes, is the release of dissolved organic carbon, which is what we hypothesize to be the stimulus for in sudden enhance of denitrification. Because what is happening, well, the literature on peat soil tells us that people have identified that the phenol oxidase exoenzyme, so essentially microbes are sending enzymes out, they're called exoenzymes, and one of which is called the phenol oxidase. And the phenol oxidase is totally inhibited under anaerobic condition, so it will not work. So that's why in swamps, organic matter just accumulates, so does it in peat soils. That's number one. And so because it doesn't work, phenolics and other molecules tend to accumulate. And phenolics, some of them are notorious antibiotics. So not only is it, so that means the carbon that's available there is in the phenolic form that is generally aromatic. And as you may know, it's not available for microbes. So first it needs to be oxidized into a form that then become a substrate. So what we think is happening is that as we create the aerobic conditions, the phenolic molecules that have accumulated into the substrate or in the soil, or in our case, the bioreactor, turns from being one antibiotic and two at least useless for microbes and or into actually a substrate. So that's why there's such an enormous stimulation. That's why I'm convinced that the combination of both the what I call hydrological hot moments and the hydrological hot moments actually create these biogeochemical hot moments. That's why they are both fundamentally totally intrinsically linked. And that's my hypothesis why, a lot of the research on the riparian buffers that's been done was mostly done by non-hydrologists. And they were mostly soil scientists who had less of interest in the hydrology part of things. So that's why they went into the soil carbon, the vegetation, the width, blah, 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 and all that stuff. But, and that might explain why actually it is the hydrology, understanding the fact that the hydrology is extremely pulsed and there's something big happening in a short amount of time. This might be why they have missed something, or at least that's my hypothesis. Yeah. And plus there were so many other things to discover. So all that to say, what did it change? What does that change during the hot moments? Yes, the DOC is the one parameter that changes suddenly and enormously. And I think this is what make everything change at once. Hopefully that was a long answer to your short, good question.
Okay. I think kind of maybe one last one here in the interest of time. They wondered if you could talk a little bit more about how the pulsing research you described points to the hot moment hypothesis. The connection was not quite clear to them.
Okay, yes. Thank you, Jane. Well, I was trying to do that just now.
Okay.
In other words, yes, again, it is the fact that the reactivity of the hydrological hot moments, that is water table rising, number one. Two, water flowing faster. These two things have, I hope I've been able to show that, they have an impact on the biogeochemical hot moments in the sense that it is during the draining phase that the phenol oxidase exoenzymes do their work. They transform something that was unpalatable for microbes to something that actually is not only palatable but very good for them. But that will not occur or not in a significant way until the water table rises and a lot of nitrate arrives at the same time. So there's a conjunction of where the nitrate come in as in lots of quantities just as the water table rises. So there's this conjunction. The pulsing of the water actually favors the pulsing of the biogeochemical activity. I hope this is somewhat clearer.
Yep. All right. I think that gets most of our questions, Francois. So I want to again thank you for being willing to help us out with the Iowa Learning Farms webinar.
My pleasure.
Also,
thank you for inviting me.
Yeah, and you know, Chad talked on the webinar not that long ago. So we, for folks that maybe didn't see it in the chat, put a link to his talk, the archived recording of that. Again, if folks need a CCA CEU, please email Alena Whitaker, ALENAW @iastate.edu by 5 o'clock today. With that, thank you very much, and please tune in next week for another webinar.